All right. Hello, everyone. Thank you guys for joining the TNT seminar series for May. As a quick intro to TNT, as I usually do, just to remind you guys about it, TNT is the Translational Neuroengineering Technologies Network at Hopkins, and we're a group for anyone, undergrads, graduate students, faculty, anyone who's interested at Hopkins in neuroengineering, wants to learn more about it, can attend our events. We have things like the seminar series, we have a journal club, we have social events, professional development events. We had a holiday party in Christmas time or in December. So if you're interested in neuroengineering, feel free to join the group. We have a mailing list, and we have a website that I'll post in the chat if you are interested in being more involved. A couple notes. As usual, we'll be recording this talk, where we have been recording, and it's going to be posted on our YouTube page. So if you don't want your video to be included, make sure to turn off your video. Also, there will be a poll at the end, kind of to get some information about who attended and your interests. So please make sure to fill that out so that we can kind of know what you guys are happy with and how you guys want to get involved and so on. So that's it for the intro for TNT. I'd like to welcome Jim DiCarlo, who will be our speaker for today. He is a professor at MIT. He is the head of the Brain and Cognitive Science departments and professor of neuroscience. He is a member of the McGovern Institute for Brain Research and the co-director for the MIT Quest for Intelligence. So he's very knowledgeable, and I think the talk is going to be exciting. He's also, as I understand, did his MD PhD at Hopkins, so he's returning to one of his alma maters today to talk to us. So we're very excited to hear from him about vision, and I think there's a lot we're going to learn over the course of the seminar. So thank you again for joining us, and I'll leave it to you to take the lead now.
Hi, Mark. Can you hear me okay? Yeah. You hear me okay, Mark? Yeah, okay. Yep. Hey, thank you for the introduction. So, and thank you all for attending. And yes, I did do my MD PhD at Hopkins. So when I was invited to give this presentation to you, I jumped at the chance because I love the place and I'm just want to give back in whatever way I can. So I thought I'd try to tune my talk today to just, again, it's sort of translational neuroscience is often how it's pitched. So we're a basic science lab at our core, but we've tried to tune my talk to try to show you how we think some of the models of vision that we're building have translational possibilities, or at least give you hints of those. So the overall flavor of my talk is going to be describing a path through models that have been built and how they relate to possible translational goals. And please, I think this is informal. I know this is a group where if anybody, I'll pause for questions, but if you have questions along the way, Mark might field them. So I'd rather usually stop and interrupt it and have questions than have me just talk at you for a whole hour. So, and I will stay after the hour if people would like to continue after that in further questions. Okay, so with that, I'm going to get started. So I titled the talk Deep Network Models of the Deep Network Mechanisms of Part of Human Visual Intelligence. And I'll show you the part that I mean in a minute. Some people have said, is that title, is it a typo or something? And I'm like, no, it's sort of an explicit acknowledgement that if you want to model something, like maybe you want to build models that look like the thing that you're trying to model. And so, and that's kind of why I phrased it that way, because the network I'm going to tell you about is a deep, complicated network that processes visual information. And we use deep models that try to approximate all levels of that system, as I'll show you in a minute. So let's sort of back up though a bit. I like to give this introduction to sort of remind you why we, the broader royal we, are in this game to begin with. So, you know, there's a human brain. So 20 watts of power or so, does all these remarkable things. And here's a quote I like from the late Francis Crick. He wrote, your joys, your sorrows, your memories, your ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. So this is really a statement of materialism that, hey, there's this machine that could be understood, and everything that we are amounts to something, this machine that's basically a collection of nerve cells. And sort of we all sort of intuitively believe this, but I want to kind of put this up against a kind of a sort of similar idea here. So here's a device, a complicated device. Many of you may have a version of this in your pockets right now. And here's another quote. Your remarkable favorite app, all its performance and amazing user interface are in fact no more than the behavior of a vast assembly of transistors. So that's also true. And the reason I'm saying this is because that's not as jarring sort of emotionally as the one on the left, but I'm saying this because it's sort of, and this is an anonymous quote. Actually, I wrote this quote. The true but not yet useful hypothesis is how I call this, right? So the thing on the left is true, but it doesn't tell you in an engineering sense how this can actually be done. So just because this is true doesn't give us any useful, let's say, translational impact or even really scientific content in this quote. It's really more of a motivational quote, if you will. So how do we turn this motivation into reality? So how do we do science here in this space broadly? What kind of science answers do we seek in these complicated neural systems of understanding questions of intelligence or perception? So the amazing phenomena of human cognition, some of which I just alluded to in that quote, they're emergent phenomena of a set of specific mechanisms, not just the brain, which is some kind of neural network, but also its interactions with the body. So somehow both of those things give rise to all the amazingness of each of us. And again, this is to echo, each of these will ultimately be understood in the same way the phenomena of like your iPhone example is understood, another engineered complex system. How are the phenomena of transistors to iPhone user interface, how are those understood? Think about that for a minute. The answers are, it's some reproducible engineered quality, i.e. transmittable and reproducible specification of how the component parts, whether they're transistors or neurons, interact to produce the phenomena, whether they're behaviors that we measure or whether they're the user interfaces, to use the app analogy. And at some level, this is obvious, but it's really a statement against the idea that the answers are going to be simple, laws of Maxwell's equations or Newtonian or something like that. There will be room for theory, but the engineered quality specifications is really what we're after, at least to get going on these kind of progress in these regimes. And I call these kind of specifications, I'll define them as system models. And I'll say a little bit more about that in a minute. So that's my definition of what we're really after, is working system models that can approximate what's going on in these systems of interest, i.e. the brain and its resulting behavior. And so the goal is really to discover and create these system models, but with the understanding that not just having those models as a scientific goal will unlock new application goals. So that's the overall thesis in which I'm positioning my talk. This is statements that are very broad about our field, but I'm now going to zoom in on kind of how we think about this and the problems that we are able to concretely work on. Just for a minute before I zoom in, I'll just remind you that having those system models, and I just said this, has all these possible ways to interact or to apply. Of course, people think about how you use them for things like artificial intelligence. If you can build it, then you could build it into a machine. New ways to educate ourselves and our children, new avenues to treat brain disorders and even augment our own brain function, and more deeply, the scientific question of understanding ourselves. So all of this revolves around having engineered level system models, which are informed by both brain sciences, like neuroscience and cognitive science and psychology. They make measurements and discovery. They're empirical sciences or so-called natural sciences at their heart. And engineering, which on the right here, which is the creation or the synthesis of things, building things, whether it's out of hardware or software that rides on top of that hardware, or bodies that are machine bodies in the case of robotics. But these are sort of attempts to create or synthesize things. And these things come together around the area of what I'm referring to as system models. So engineering things that are trying to be models of the biology on the left. All right, so the way I like to say this is account for each ability of the mind ultimately is a goal, using the systems of components of the brain, which for us is connected sets of neurons, in the language of engineering. Hopefully this is not a bit of it's not objectionable at some level, but I've noticed that this frame that we take here is not a common frame of how people think about what understanding even looks like in these kinds of systems. So today I'm going to review for you an example of this, again, in the area of visual intelligence, which I'll call perception, and not all of visual intelligence. And I'll sort of again explain to you why this matters. So for system models, what we're after is specific neural network models that can actually perform and accurately explain the empirical measurements from brain and cognitive sciences. And I hope that becomes clear as I go through the talk. Now, when I say we, it's the broad field that I'm interested in describing here, but I'm going to talk mostly about work from our own group. And this is our lab team and just the folks that I will try to highlight along the way, their contributions to specific things, but putting this in a broader context here. And all the credit for anything that you see scientifically really accrues to these amazing women and men. And I'm really just an ambassador of that work. So the outline of what I wanted to try to tell you today is basically these 5 points, and I'll try to go through them at a reasonable pace to give you an overview. So I'll give you some background of like what are our leading models of the visual processing. I'll then kind of tell you how those models are being continually upgraded and how that process is going and being managed, or give you some sense of that. Then I'm going to shift more into this application mode I alluded to at the beginning. It's like, how can they start to be utilized to sort of deployed to do things of interest to us? And then I'm going to talk about kind of even coming back to deploying them in a way of exploring new biology. And I'll show you a recent example of that from our group, which then emphasizes a kind of a speculative application that goes, combines areas 3 and 4 to say, how might we use these models to unlock new ways to modulate brain states and maybe ultimately improve human well-being? And that will really just be a kind of speculation at the end to motivate some discussion. And I hope draw on some interest from all of you interested in neuroengineering broadly. Okay. And Mark, please tell me if anybody's got questions along the way. Okay, so background. How are visual system? How has our field developed models that really around 2014 were sort of starting to take off as models, the leading models of the ventral stream? And so let me just sort of give you context here. So my group works primarily on something called visual intelligence or perception, which is questions like when you look at a scene like this one, how do you tell what's out there? How can you be able to say things like, what's in this scene? Where are the cars? Where are the people? What will happen next? Where is it safe to walk? More advanced levels of visual intelligence. Notice the ones on the left is just what's out there, and the ones on the right are a bit more about predicting the future. And these are kind of questions that motivate people in both computer vision and AI broadly. And they also motivated the initial work in our group here. And we think we've made pretty good progress as a field on the things on the left, like knowing what's out there at all and models of how the visual system does that. So in the intro of my talk, when I said part of visual intelligence, this is what I mean by part of. We don't have all of these questions of visual intelligence, but we're on this path towards answering them. So I'm going to tell you about the sort of early steps on this path to understanding these kind of what's out there questions. And that path begins by you thinking about first as a biological organism, when you look at a scene like that, when you don't actually have a high acuity map of the whole scene at once, you really have high acuity where you happen to be foveating, say this is your center of gaze at this red dot. And I'm going to talk about how the brain processes the central 10 degrees or so, which is illustrated by this circle here. And the way that you take in that scene, when I just showed it to you, and you didn't really probably know that you were doing this consciously, but what your brain was doing was busy moving your eyes around when I put up that slide to say, what's in this scene that Jim's putting up on that slide? Oh, there's a person over here, there's a sign here, there's a car here. So you're making these eye movements called saccades, that are rapid eye movements, and these fixations where you dwell for just a couple hundred milliseconds and gather information, high acuity information. Hopefully those of you, this is background for many of you, but just kind of allows us to operationalize the problem of not just digesting that whole scene at once, which we don't do, but digesting components of that scene as we make eye movements. So just a couple hundred millisecond glimpse at each fixation position is where we started a lot of our work. So we define, here's examples of some snapshots of 10-degree snapshots out of that image, or trying to approximate 10-degree snapshots, which are showing you really one dominant object in each and every one of these snapshots, a dominant car, a person, a sign, and your ability to extract that there's a car in this snapshot, there's a person in this snapshot, there's a scene, there's a sign in this snapshot in just a couple hundred milliseconds. Here I'm showing around 200 milliseconds. That ability we call core recognition, to just be able to do that. Again, a part of visual intelligence. And so that's the part that we've spent a lot of time working on. Again, I want to emphasize this is not the end of the vision processing. It's just a way to get started in modeling these system models. And so here's how it works in a lab. If I wanted to test you in how well you're able to do this, if you fixate that dot and I say, here comes an image, get ready, and then an image came up, I didn't pre-cue you as the kinds of things I would show you, other than saying maybe the word object. And hopefully you would notice that it's more likely the object on the right than the thing on the left. We'll try it again, more likely the thing on the left than the thing on the right. And these are ways that we can kind of gauge your ability to judge what's out there, even in complex scenes. That was a natural photograph, again, hopefully the one on the right and not the left. Okay, we can get lots of data like that from humans and also from our cousins here. This is what we mostly do in our lab. This is the rhesus monkey doing the same task that I just asked you to do. He's triggering the presentation of a test image and then choosing among two choice images. Again, it's not just one object. This animal knows, I think, on the order of 50 objects or so. And and the animals rapidly learn new objects, and then they can generalize their ability to identify those objects in complex images very quickly. They learn that in a day or two for each new object. And again, they can do this all in an interleaved mode. So there's no attentional pre-cueing in terms of what kinds of objects, just spatial attentional cueing to just the center of gaze, see the image for a couple hundred milliseconds, and then make a choice. And again, these primates like us are actually very good. These are patterns of performance across monkeys on the right and us on the left. Both of us are primates, just different types of primates. And what I'm showing you here is sort of D prime values. And I don't want to take you through the details of how the slide gets created. I just want you to know that these are sort of patterns of difficulty across many different types of objects, which you see listed on the left. And the main takeaway is notice how similar the pattern is on the left and the right. In fact, they're quantitatively almost very difficult. They can barely be distinguished. So they're not quantitatively exact, but they're very close between these two species. And again, that shouldn't be surprising. We have common evolutionary roots, but it says that the rhesus monkey is a very good model of our visual system and doing these kinds of tasks. And so the way you should think about the rest of the talk is I'm going to treat rhesus monkeys and humans as essentially equivalent and how they handle these kind of intelligence problem or this visual processing problem. And at the time, computer vision systems were not, when we started this work, were not nearly as good as us in terms of average performance. They've now approached us with some of the modeling advances that I'll tell you about. They're still not equal to us in terms of their patterns of performance over images like the ones I was just showing you. So it's not just saying you're good or bad at something, it's the patterns of how you're good or bad at something that we mostly track at the behavioral level. It's sort of both an absolute performance and the patterns. Okay, so the fact that monkeys do this quite well and similar to us quantitatively gives us license to treat it as a quantitatively good model of the humans. Instead of now studying the human directly, we can study the rhesus monkey brain directly. And the reason this is powerful is we know a lot more about the rhesus monkey brain than we do the human brain, and we can approach it with tools that we can't easily use in the human brain. We know, for instance, in the monkey that this processing is carried out in a series of areas called the ventral visual stream, here shown in different colors. These are the main cortical areas that are involved. Here they are unfolded in sort of engineering terms, where we think of the millions of neurons schematically illustrated in each area. When I show you this slide at the bottom with these different areas, it's meant to just schematize the actual neurons in the biology, even though it looks like an engineering diagram. This is really the brain spec, if you will. And this is roughly the feed-forward architecture that we know of the ventral visual stream, with inputs coming in on the left and processing gradually up to the right. And it takes about 100 milliseconds or so, and it also has both feed-forward and recurrent processing motifs that are illustrated by these arrows. Okay, we know this ventral visual stream is involved. We know this because of lesions in the monkey brain produce deficits, also similar lesions in humans. And you can think of this area again as processing images like the ones I'm showing you here on the left, and processing them to a form that allows us to support tasks like visual recognition. I'll show you that in a second. So this complicated network, this system of architecture that we can see neurons firing all up and down the system when you show images like these ones, how do we aim to scientifically understand this system? How do we scientifically proceed? Well, this is what I said at the beginning of the talk, is we try to create so-called, I just skipped a slide there, system models that are meant to approximate what's going on in this kind of network. So when an image like this one is shown, it produces a pattern of activity on your retinal ganglion cells, which is essentially an isomorphic copy of what's out in the world. Think of your retina as like a nice camera translating the photons into neural activity patterns that are transmitted down your optic nerve to your lateral nucleus in your thalamus, the second stage here, which are then transmitted to visual area V1, these cortical areas you see at the top, and these series of cortical areas V2, V4, and this sort of high-level cortical area called IT that I'll say a lot about. IT stands for infratemporal cortex. And again, you can think of each image as producing a population pattern of activity in IT cortex. If you show a new pattern of activity, it produces a new image, it shows a new pattern of activity in IT cortex. And if you cycle back, and IT can go ahead and respond even quite readily at the rates of these images shown here, with a lag of around 100 milliseconds. So the throughput of this system is quite good, and it can easily keep up with images at rates even faster than your natural saccadic eye movement rate that I mentioned earlier, again, around 200 milliseconds per fixation. Okay, so that's by way of sort of context of how we approach this particular biological neural network. This is an actual monkey looking at a screen. This is an image of a monkey looking at a screen. Conceptually, the reason I'm showing you this is because to orient you to the kinds of data that we can get out of this visual system in the monkey, not just the behavioral data I showed you earlier, but now the neural data from the different processing levels. This is an example of like a single unit recording in IT cortex. Here's the spikes you might get shown at the top out of one example neuron in IT cortex to 4 example response images. I'm just showing you this to bring you down to the most elemental level of the neural physiological data, which are the spikes out of individual neurons that you see here as these blue tick marks. And this is, again, I'm going to zoom out of this in a minute, but just to give you a sense of what these neurons do, each row is a different trial, different presentation of this image. And here's 4, and notice that this top first site one likes the first image and the second image. It gives a lot of spikes out of that images, again, with a latency of around 100 milliseconds. You see the time scale at the bottom. Doesn't like the second and the third image, or the third and the fourth image so much. Site 2 prefers the second image and maybe a bit the third and the fourth, and site 3 likes the third and the fourth images. Okay, this is just to show you that different sites have different preferences over arbitrary sets of images. These are just 4 arbitrary images that we happen to test in this particular study. And our goal has really been to understand these spiking patterns here. Now, what we do in most of our work is not to try to model individual spikes and their time at the millisecond resolution, but we instead are modeling the mean spike rates that occur in time windows like these ones shown here. This is a 100 millisecond time window, and so you can think of just averaging how many spikes are in that big block there. And you could say now all those spikes that turn into look like individual millisecond times, I'm just going to count as a mean rate. 60 spikes per second, 71 spikes per second, 25 spikes per second. So there's each image producing a mean rate out of each neural site. And those are the elemental data that are most motivating to us. And I'll say more about why that is in a minute, but that's the level with resolution of the models that I'll talk about as well. So they're trying to model mean rates of neurons. Again, think about 100 millisecond time windows, and we've taken this down to 10 millisecond time windows in some of our more recent work, if you'd like to ask about that in questions. Okay, so back to the big picture here, you show a bunch of images, you get a bunch of spike patterns out of IT cortex. Here they are now zooming in on IT cortex. We don't just record one neuron at a time, but we can record hundreds of neurons at a time with these multi-electrode arrays that we chronically implant during an aseptic surgery. Then we can have animals connect every day, record over many days. Neurons of these electrodes, at least the multi-unit activity is very stable over often many weeks or often even months to be able to record single sites. And then we can go ahead and build these like larger spike feature vectors at the single site or multi-unit site resolution, where we now are showing you the response of many neurons. If we pool across animals, we can get thousands of units. So now this is an example of how thousands of units respond to this one particular image here shown at the top. The green color indicates the mean spike rate, the sort of thing I just mentioned a minute ago. High spike rate means high green or full green. Low spike rate shown in black is just black. And what I'm just trying to illustrate for you here is there's like a pattern that happens. This is an empirical measurement for that particular image. So we're going from this conceptual idea of the left to an actual measured pattern out of IT cortex. Okay, we don't, of course, just record one image. Here's now 7 other images. Notice that you show different images, you get different patterns. This is in line with the conceptual idea I've been telling you. And then here's thousands of images. Again, we're recording many thousands of images over a week or so in this particular data set, just to give you a sense of the kind of data volumes that we and others can collect using these methods. Okay, so I'm telling you all this because you have these spiking neural patterns out of IT cortex at the top of this deep visual hierarchy. And we know from prior work that I won't be able to take you through in detail is that this neural population representation is a very good thing to be trying to model. Why do we say that? Because when we build simple linear decoders on top of this, train on a few examples of objects, just like the monkey has to learn a few examples, it generalizes to predict the animal's behavioral patterns even over new images of those objects. And so that's called a linear decoder, and it produces this kind of behavior that matches the human. And that's why we think of this as a sort of good start point. I think I heard some questions perhaps, or maybe that was just noise. Let me just pause here, Mark, to see if there are, I'm going to reach a pause point in a minute, but let me just go one more second and then I'll come back to it. So basically I'm giving you some context here that the top of the ventral stream produces spike patterns. I'm empirically showing you to them here. We have decoder models of how these spike patterns relate to the animal's behavior. This leads to BMI kind of questions, like if you silence the neurons or activate those neurons, that should lead to changes in perceptual behavior, so direct injection kind of translational things. We've explored some of those. Those are hard things to do, and be happy to talk about that. I'm not going to talk about that for the rest of the talk, other than to say those kind of decoder models allow you to start to answer those kinds of questions. But let me just, most of my talk, I want to talk mostly about what's happening up to IT cortex, not from IT cortex to behavior. But the way I want to kind of position this is you should have now in your mind that if we can build models that explain the mean firing patterns out of IT, then you've got a pretty good model of what's happening end to end from the image all the way up through these different levels of neural processing, all the way up through simple linear decoder models that can then predict the behavior quite accurately. So that's been the frame we've been pursuing this. And I'm going to sort of like pause here and see if there are questions about that overarching frame before I dive into the network models of the ventral stream. Okay, Mark, I'm not hearing anything, so I'm going to keep going. All right.
Yeah, go ahead.
So, okay. So, again, I put a check mark here. This is not a finished thing. Nothing in science is ever finished, but we have some decent models. So let's move on to like, how is this computed? So I'm saying there's these powerful patterns in IT cortex. How are they computed out of this image that comes in the front? So, and then what is the point of that? So, okay, so just some fence posting here for all of you. This is a how is being done question. This is a question of adult online mechanism of so-called inference. I'm not trying to talk about learning. So I'm not trying to do how evolution occurred or how the networks get learned. We're just trying to, by hook or crook, any way we can, find a good system model can approximate what's going on from the images, through all these different neural levels up to IT cortex. That's our goal. Find, build, acquire any way we can an accurate, again, systems level model of this. So please take everything I'm going to say next as really in search of that goal. Those other goals are interesting, but that's not the goal that we've been after. So how do we proceed in doing this? Again, we need to find a system model, back to what I said at the beginning. Okay, I'm going to skip through this quickly. The kind of system models we build are basically artificial neural network models. For those of you who don't know, the way an artificial neural network model works is it basically has as a unit, a simple approximation of a neuron, which takes a bunch of inputs, has a simple summation shown here at the bottom, some weighted summation with some nonlinear output function here with the sigmoid, that sigma, which gets transmitted to other neurons downstream. That's one model neuron. You might have many neurons talking to each other. Maybe they live in a particular area, like a particular brain area, like V1. And if you want to model the whole system, you're going to need multiple brain areas. I just showed you on the ventral visual stream. So the images might come in on the left. There's some processing that might be feed forward, leads up to behavior, as I said. There might be feedback processing as well. There could be cross area processing in addition to that. The reason I'm telling you all this is basically just saying there's a big, large family of artificial neural networks as a family of models that really is our starting point as neuroscientists for modeling brain function and behavior. So that's the family of models that we are strongly committed to as neuroscientists, accepting the fact that this is only an approximation model of the neuron, and every model we'll build on the right is also going to be an approximation model. But if we can make them better and better, ever more accurate, we can start to make gains on these problems. So just to orient you, the parameters that you need to do when you build neural network models, for those of you who don't know, is you have to specify the so-called architecture, which is what's sort of shown on the right. And you also need to then, once you have the architecture, imagine all these units here, perhaps millions of units living inside these models where you have to tune up the weights of how neurons connect to each other, which we think of as the synaptic connection weights, or at least approximating those synaptic connection weights. Okay, and you may have heard of convolutional artificial neural network models. Those just are simply models that respect a certain layout of how the sensory inputs are processed. And hopefully that'll become clear in the next few slides. But for those of you who don't know, convolutional deep neural networks is what I'm describing here. And that's a subfamily of the larger class of artificial neural network models. And when I say model, again, just to orient you, a model for us does not exist until all the parameters have chosen. Before that, it's just a model family. It could be anything. You tune up the parameters. But once you freeze the network itself and all its parameters, now you have a model and it can make predictions and comparisons to brains. And those are the kinds of models we use to make our comparisons. So this is not a new idea of building ANN models of how to model the visual system and how you go from the sensory inputs to the processing, to the final neural stages and the behavioral outputs that I described. This idea actually goes back to at least the 1980s. One of the famous models was Fukushima. This was followed by other models you might know, those who work on vision and V1-like and V2-like models that model the early parts, higher level models as shown here at the top. You don't need to know all the details of these models. The only thing you want to know is like, well, if these are good models, they should actually align well with the internal measurements of the brain of the type I was just showing you here. They should align with the natural sciences data. So if you go ahead and record, for instance, an IT neuron, and you look at its response over many images, which is what I'm showing you here. Now the y-axis is the response to different images. This is about 1,600 images. Here's 4 example images. Notice that there's this complicated pattern of responses. I grouped them by category in case you think these are category neurons. I hope you look at this and say, well, squint at it, it might be category-like, but it's not. It's clearly not exactly category-like. It's a complex pattern of response. Here's a area visual V4 of another example neuron. I'm showing you, again, complicated with respect to category, but clearly some reproducible variance here. And I didn't show you the error bars on these points here, but they're quite small here. So these are the mean rates over many images. And I'm showing you those because these are the kind of primary data from the neuroscience side that we'd like a model to be able to accurately explain, not just fit, but predict in held out situations. And this is kind of a long-standing goal in our field to be able to do this. And these first generation of ANN models, they were actually not very good at actually explaining these kinds of data. And the way we can quantify that is here on the y-axis. This is the fraction of visually driven neural response variance from 0 to 100%. And the way that this gets quantified, and this is a bit of a technical detail, but just so you understand, when you record from an individual neuron in a brain, you have to have it mapped to a set of neurons in a model, or a specific neuron in a model. And the way that's done is by taking some data to draw a linear mapping from neurons in the model to individual neurons that you recorded in the brain. Then you test on some held out data to say how good is the prediction on that response of that neuron on some held out images, for example. And that's what's essentially on these plots here, is a sort of a mean over many recorded neurons, in this case, visual area V4. And so you can take this as a summary metric or a summary benchmark of how good these models shown at the bottom are explaining neural response patterns in cortical area V4, which again is a mid-level visual area in the ventral visual stream that I introduced you to. So again, I just said this, explain means predict for new images, and this accounts for noise limits that exist in the data sets. And so this 100% is already noise ceiling. So this is how you should think about this plot. And back to the big picture here is that once you quantify these things, you can see that the models at the time, and here, even the best ones, they're not very good at explaining what we're observing, even accounting for noise, what we're observing within the brain, the visual system itself. And you can see here in red line is the prediction of this model trying to predict this response pattern of this V4 neuron. And you can see it doesn't look very visually very good. And that's what's quantified on the left. If you go up to the next higher level area IT that V4 feeds, you see the predictions, not surprisingly, look even slightly worse than they do in area V4. Again, poor brain alignment of this first generation of ANN models. So this was, motivating, says these models are not good enough, so we need better models. The second generation of ANN models, as I'm referring to them here, are, again, they look like the first generation. They're deep artificial neural networks where you have neurons inside of here. You've got convolutional operators in a sense approximating what's happening in parallel in the brain. In the visual system, that's textbook visual neuroscience. And you've got these 4 different areas here, meant to model the areas of the ventral visual stream. And so again, this is what I just said. These are still, when I say still, that model family has been around since the 1980s. This is a deep artificial convolutional neural network. What changed more recently is that people have started to be able to model the actual goal of this system and to implement these models in a way that allows them to perform. So we call this a performance optimization approach, where you choose a task like do core recognition. Say there's a car here, a person here, a sign, the core recognition task I introduced you to at the beginning. And then you use engineering as a tool to optimize all those free weight parameters and even the architectural parameters, some of them within these models, to optimize it to just do well at this task. That was essentially the game that we played a number of years ago. This is especially Dan Yeamans, who's now at Stanford as a professor, and Ha Hung, who was a graduate student in my lab at the time. And they played this game of let's optimize networks to do well on core recognition, freeze the parameters, and then compare those networks with the brain. So we
have a question.
Yeah, okay, let me pause here, Mark. Go ahead.
All right, so the question is, can you talk through a bit more how you do the mapping between the brain neurons and the ANN/DNN models? My very limited understanding of brain neurons are that they work differently, depend on timing between spikes, whereas DNN/ANNs are not spiking.
Yes, so, and I'll go back to that's a great question. Let me go back to what I said. One thing, we're only trying to model the mean rates. So in that sense, we've abstracted away the spike timing details of neurons. We start treating them as activation values. So they're not zeros and ones now. They're just a mean rate that goes from 0 to whatever the maximal rate, say, have 200 spikes per second might be. And we're trying to find models that can approximate the mean rate. So we're sort of averaging over repetitions of images. And we're doing it within specific time windows that I said earlier. So that allows us to get away from a lot of the sort of more subtle hardware biology. We're sort of ignoring that when we build these kinds of models. And that could still be important. And so that's why I'm calling it all out. That's how we're modeling things right now. And then part of this question is I said in the sense of, hey, I've got a bunch of neurons in a model that's sort of at the top that have activation values. So you can think of them as mean activation values in response to any image. And I've got neurons at the bottom that also have mean rates, the biological neurons. So I have to find a mapping between the neurons at the top and the neurons at the bottom to make a prediction. How do I map a model neuron to a brain neuron? And again, the way we do that is to gather a bunch of images that you show to both systems, just like I'm showing you here. Here's some images coming in the left. You show the model the images, you show the brain the images, and then you say, okay, let's go to visual area IT, for example. I think I may have that on the next slide. So IT, compare IT to IT. So you have an area called IT in the model, an area called IT in the brain. You just take the neurons within IT as a basis set. You regress onto the individual recorded neuron in IT. So by regression, I mean you take the data from the IT neuron itself, its mean rate. You have a linear regression on that IT basis set, and you often have to have some regularization when you build that regression. Then you test the predictions of that regression for that recorded neuron on held out images that you did not use to construct the regression. So we call that the mapping step between the model basis and the neurons recorded themselves. I hope that, and then so now you have a sort of alignment between models and brains, and you can get quantitative about how well it predicts in that mean rate framing. So I hope that answers the question, but again, of course, the biology it's a model. It doesn't respect all the biology. It's not trying to model all the biology, just the mean rates. And please, if the question's still there, I'm happy to keep going, because again, this is for you guys.
So thank you. So thank you.
Okay. All right. So then, okay, the big picture here is we have models at the top, we have brains at the bottom, we can compare them in the way we just described. And the big science advance was that, hey, we started to see impressive matches between these ANN models. And again, these new ANN models, these particular ANN models have been around for a long time, but these particular ANN models started to match much better than the old first generation models in the way that I just described for you. And here, let me show you that visually. Here's that same neural response I showed you earlier. Now here's the prediction. Look at how the red is the prediction of this match between the model IT and this example IT neural site. It's just one example, so you can sort of get intuition of what's going on. Notice this neuron in black has this very detailed pattern of response over the images. And even in red, the model seems to follow that very precisely. Not exactly, though. We can still see it's not perfect, but it's much better than that earlier model I showed you a minute ago. And when you quantify this, it explains about half of the explainable response variance. So it's still a glass half full story, but it's a huge advance over the kinds of models we had before. So they already start to we already seen these kinds of gains. And the rest of the advances may require, again, more attention to the details of the biology and the question that just came up. But the thing you should take from this is like there were big gains in the ability of models to model individual biological neurons in this manner. So this is what I just said here. We have a kind of glass half full, half empty story. The half full story is this, models have become much more accurate at modeling the internal mechanisms and the supported behaviors. But again, half empty, that we can see that they don't yet explain everything. Okay. In the interest of time, I'm going to just sort of zoom through a couple sections here. That was just a general idea. I gave you context of our problem. I sort of gave you sort of how those models came from. And I showed you an example of one model fitting the data. There's not just one ANN model of the ventral visual stream. These models continue to evolve to have more and more accurate models of the ventral stream, getting upgraded all the time in a way. And one of the things that our group has been trying to help organize in the field is a tool for doing this, a platform called BrainScore, which tracks how well different ANN models are aligning with many levels of the visual system, which is shown here on the right. This is, again, a number of people working on this, especially Martin Schrimpf, who was really the leader in this effort. If you're interested in the kind of all the philosophical and other technical issues behind that, I would point you to this particular paper here. I'd just like you to say that this is a platform that continues to grow, and it's a platform for the field. And both experimentalists and modelers are adding models on the left, and experimentalists are trying to add benchmarks on the top. And that's allowing us to sort of, as a field, start to converge around kind of, are models getting better? What are they getting better on? What are they getting worse on as the field progresses? We actually ran a competition recently that drew in a lot of new models, and actually we found models that were even better than the models that we had been using at the time from the community, trying to compete on trying to achieve these kinds of gains. In the interest of time, I won't take you through all that, other than give you this sort of idea that the field is now building more and more models that have better and better alignment empirically assessed with the visual system. And so that platform of brain score is really just contributing this larger idea introduced at the beginning of trying to find better and better models as they align with the brain and behavioral results. I don't just mean predict IT neurons. I mean predict IT and V4 and V2 and V1 and the behavior. That's what better means to us, is trying to do all of that and trying to engineer those kind of models. And this is a never-ending loop that you see here in the middle. Better models lead to better alignment with the brain, but not perfect alignment, build better models. And this goes back and forth as sort of good science does, if you will. What I want to spend the rest of this talk talking about is like, okay, let's accept that the models are being built. They're better models of the visual system. And Jim is saying we should call these the models the current best scientific hypotheses. That's the thing I'm putting forward to you today. Now, if you accept that idea that those are the current best scientific hypotheses, you say, well, okay, what can we start to do with these hypotheses? There's a debate in our field. There's like, do these kind of models, are we just building digital copies of the brain, these ANN models? Are they just simulations? Does it really count as an understanding? There's a sort of philosophical debate going on. We've sort of engaged a little bit in that debate, but in the way we've most engaged in it, instead of sort of trying to have a philosophical debate, we've been just asking, as engineers, given that we have these models now in our computers, what can we do with them that we couldn't do before we had them? And so what can we do with this kind of understanding? And we hope this is the part, I hope this is the part that most resonates with this group here. It's like how we think about deploying these as engineering tools. So as an example, I'm going to show you a couple things that we've been doing with these models in the last part of this talk. An example of how these more accurate AI models can be utilized. So just to give you a big picture here, I'm generally thinking about things that might be called applications. So that's a very gray area between basic science and applications. One is computer vision and AI. Unless you've been under a rock for the last 5 or 6 years, these kinds of deep network models are now taken over computer vision. They've actually taken over other areas of AI. And they really were inspired by these kind of visual networks that I'm showing you here, but they've gone off in all kinds of directions that way. I'm not going to talk more about that today. They could also be used for guiding visual learning. We've done some work on that, and I'm not going to talk about that today. But what I am going to talk about is their use in neural control, and they could be used in brain-machine interfaces. I'm not going to talk about that today, but these neural control and brain-machine interface ultimately cash out as human health applications, at least that's how I map these things. So the rest of this talk, I'm just going to talk about an application called neural control of using these models and show you what we mean by that, and then speculate as to kind of where that might be headed with these kinds of models. Okay, for us, this began with this paper. These are 2 postdocs in the lab, Puya Bashivan, who's now at McGill, and Kohij Kar, who's setting up his own lab now at York University. And so when they're in my group, they did this particular work about 3 years ago, which we call neural population control. And the basic idea was to test this idea of using these models as tools for modulating the brain. The way this game works goes like this. So you have a model of the visual system shown at the top. This is like one of the ANN models I'm showing you. It's just a different format of the drawing at the top. You have the actual brain at the bottom. And the basic idea is you say, look, I'm a neuroscientist or I'm a brain scientist, and I really want to modulate area V4 in a very particular way. For whatever reason you have in mind, maybe it's to improve something about a brain state in an actual application, maybe it's a neuroscientific goal, but I want to modulate V4 in a particular way, and here's the way I want to do it. So that's the kind of hand of God declaration from the neuroscientist. Says, okay, now what can we use these models to help us? Well, given that we have models that go from pixels up to V4, we can invert back through the models, which is what this red is. We can now, in effect, guided search through image space using the models to guide us to find new images that can modulate the V4 activity in the ways that the neuroscientists desire that to happen. And so then this is an example of that optimization process happening where you start from a random image, use the model to guide you to something that's going to activate a particular V4 neuron. Okay, that's just to give you an example of like trying to design an image and the intuition behind that. And in this paper, what we demonstrated is that we can use, I call these sometimes digital treatments, because we think of these as sort of interventions on the system to modulate neural activity, given that we now have a model to tell us how to do that, which we didn't have before. And let me sort of show you kind of how that generally works and how well it works. And you're going to see it's a glass half full, half empty story. So the idea, again, is you have model predicted firing rate on the x-axis and model measured neural firing rate on the y-axis. Each dot is an image. And these are different test images, and you might be, first of all, wow, that's pretty cool. The model actually predicts the neural firing rate. Look how the points are all on the diagonal line. You shouldn't be surprised by that, because that's essentially what I've been telling you the first part of the talk, is we now have these really good models to accurately approximate what's happening in the visual stream, including visual area V4. So that's the earlier work I outlined for you, just shown visually in another format. Okay, now though, what we're doing is essentially trying to go beyond that to use the model to say extrapolate out to here and say, well, could we come up with an image where the model thinks, oh, now I'm going to really activate this neuron? Is it just a sort of a toy example here? We call this the super activate goal. Could we find an image that goes way out here and activates the neuron? Well, if we have the right model, we should be able to do that. And so we're going to try to super activate this particular V4 neuron. We do this inverting of the model trick I just showed you. Here are 5 example images that this particular model came up to activate this particular V4 neuron. And then it said, oh, you should now be up in this corner here is where I'm predicting these images should drive you. That's its prediction on the x-axis. And then we went ahead and measured the response of this neuron that we're recording from on the y-axis, and we found this. So look, these red points from the out, they're well above any of the blue points we have measured. So we got this neuron to fire further higher than any images we've been able to find on it using our own sort of just random sample techniques. And so in that sense, it's a success. But as an engineer, I view this as a partial failure, because these points are not on the diagonal line, which again tells us the model's not perfect. So the model lets us modulate, but not yet to the degree that we want to, but clear gains over what we had before. What gets me really excited is not just that toy example of modulating a particular neuron, but think about modulating a whole population state of neurons. So our perception is driven not by individual neurons, but by collections of neurons. So here's now a V4 recorded population. This is now 38 V4 neurons recorded simultaneously in parallel. And the control goal here, down from the neuroscientist, in this case us, was to say, could we just activate neurosite 12? I want to turn on site 12. Don't ask why, I just decided I want to do that. I want all the other sites held at baseline. Please find me an image that does that. Let me turn it over to you, Mr. Model. Mr. Model, come up with an image that will do that. And here you go to tell you this is not a trivial thing to do. This is the visual field of area V of this particular sites that we're recording. These circles are the receptive fields of these V4 neurons. For those of you who are vision neuroscientists, you know that those are the areas where the neurons respond to. So they're all responding to light energy in roughly this part of the visual field. So it's not a trivial example, just putting light somewhere in your visual field. You have to structure that light in a particular way. If you just search through images that we had to find the best image that we could find, it found this particular image, which activates site 12, but unfortunately activates all the other sites a reasonable amount as well. So it doesn't achieve that control goal in this very one hot kind of a way that I've sort of phrased the goal here at the bottom. One site active and the other's not. And so, okay, let's let the model give it attempt rather than us and our poor man's attempt. The model says, here's the image I would really like you to show. This is the best it came up with when we searched through this model at the time. Notice this image looks nothing like a natural image. It's not a chair or anything like that, although it looks like there's some sort of visual features that feel familiar with what you're seeing in the chair. The dashed red line is the neuron's receptive field, again, for the aficionados, by the way. This model decided to put energy as shown here in this lower image at the bottom. And here's what actually happened when we showed this image to this set of neurons, again, 38 neurons. We actually activated site 12, great, but we much lowered the responses of all the other sites. So we found an image that was able to much better achieve our control goal than we were able to achieve without that model. And so again, this is half full. If this was perfect, all these red lines would be at 0 and this middle red line would be high as shown here. We're not yet there, glass half full, glass half empty. All right, but this is quite cool that we're now able to start to be able to modulate internal brain states much more precisely than we were able to do before we had these models. All right, so I'm going to just, in the last 10 minutes here, I'm going to sort of tell you sort of 2 more things if I have time. So look, those were models that were 3 years old. Now we've been going more recently into this kind of next example. We're able to start to ask. Let me sort of motivate this a little bit more. So take that neural control goal, keep it in mind because I'm going to return to it in about 5 minutes when I get to the conclusion of this talk. What I want to show you here is a little bit of it might feel like a tangent at first, but it's going to come back to the thing I just mentioned a minute ago. So those of you who know about deep network models for vision will know they're subject to so-called adversarial attack, which means you can take an image like this image of a dog shown here, you can add a perturbation to it, which is shown in the middle, and you produce this image on the right. Now, this is called adversarial attack because the perturbation image is constructed to be an adversary against the model to try to flip the model's category output. So that here the model correctly says terrier, the computer vision developer is happy. Now the model, the output of the model says church, and then the computer vision developer is unhappy, okay? And this is kind of, okay, this is bad. Why is this bad? The reason this is usually thought to be bad, and again, it's called adversarial attack, brain and cognitive scientists and computer vision developers, sort of armchair brain and cognitive scientists, look at this and say, well, obviously our brains don't do this. So that's a problem with the model. We need a better model. Now, there's probably some truth to that statement, because you look at this and go, it looks like the same image to you, even though it's not technically the same image. And so you don't flip your category output when you look at this. And so some people would say, clearly, these are not good models of the brain. But we said, instead, wait, we're taking these models seriously. They are our leading models. We know they're not perfect, but let's at least ask this question of how much of this is actually happening within the brain when you subject it to adversarial attack. Does the brain even show any of these kinds of limitations at all? Now, keep in mind, again, this perturbation is not random. So we couldn't ask this question before experimentally, before having these models. But now that we have these models, we can use it to ask this sort of perturbation level questions. And again, this is what I just said here. Because we take these models seriously, we should take their predictions seriously. And we do this because it might point to more accurate models, and it might also show us some cool new biology. In this case, I think it does both. And in fact, I'm going to point to the cool new biology is mostly what I'm going to point at next. And when I say we did this, I mean Chong Guo, who's a postdoc in the lab. And what Chong did is his aim was to compare the worst case sensitivity. So to do small perturbations and look at the worst case sensitivity of individual neurons, in this case, an area IT, that first area I showed you about, high level visual area, with the corresponding high level visual areas of these models. Knowing that those models are quite brittle in the way that they produce those kind of adversarial attacks, we just like quantitatively compare models to biological IT neurons to model neurons one by one, and ask the question, which is more sensitive to the image adversarial attacks? The feeling in the field is, of course, that the models are more sensitive because we don't behaviorally make those kinds of mistakes. But we didn't know this answer to this question at the neural level. And again, these are our best models, so we sort of took them seriously and asked this question. I'm going to skip through a bunch of experimental detail to just show you the main result here. So what you see on this plot, this is the result of this comparison, and I'll try to unpack what it means in a minute. The x-axis is the amount of perturbation that we're going to inject in the system. So how much noise effectively we're allowed to have in that kind of fuzzy, that noise image I showed you. Remember, it's structured noise. And the y-axis is the adversarial sensitivity. So how sensitive, how much the response of the unit moves from its response to the so-called clean image when you add that perturbation. So flipping the terrier to the church, how much does the neuron move as you make the terrier into the church causing image? And so the old model neurons were measured, they had a very high level sensitivity here. This is a log scale, notice on the y-axis. Newer models, which are actually our newest models of the ventral visual stream, these are so-called adversarially trained networks for those aficionados in the room. They're actually our current best models of the visual system, and they have made remarkable gains. These are gains that are due to folks like Alexander Madre and others at building these kinds of newer models. They have gains in the sensitivity here. When we started to compare to the biological neurons, here's the take-home result that I led you up to. The biological neurons, individual neurons, have almost the same sensitivity, maybe even slightly more sensitivity than the model neurons within the adversarial trained networks. But they're sort of at least in the right order of magnitude now that we have these newer models. These things are very comparable. So if I show you this plot, it's very hard to sort of reject the model in terms of being more sensitive than the biological neurons. If anything, the result leans the opposite way. That's a very important result from thinking about how these models, how could we make models more robust or not? It means maybe don't look to biology in this particular case to think of where the robustness comes from. But I want to kind of turn to the fact that this is what I just said a minute ago, but I want to remind you, these are very small perturbations. Here they are perceptually. You can barely see that we're modulating these images when I show them to you as you go from clean, adding perturbations from left to right. Yet the neural response is modulating a lot. And I'm going to unpack that for you on the next slide so you can better understand it. This dashed line right here is the normal operating range of the neuron, its normal category range, and I'll show you that in a minute. And again, these small perturbations are really modulating the neural response. So this we think is a real opportunity to start to make brain network modulations using this kind of neural control approach. Let me show you this. I think it's a little easier to see. Rather than showing the results slide, show an example neuron. So this is an example IT neural site. has a high, it prefers one category over another. That category preference is defined because it gives a lot of spikes in category 1, which you see at the top. Notice, look at the part in the red box, versus less spikes for the non-preferred category when we show these images. So you can see that, like I showed you in the very early slides of this talk, the red is much lower than, or much higher than the blue. Now we're going to go ahead and perturb. This is the non-preferred category. A neuroscientist would say this neuron does not like dogs. So it doesn't like dogs. That's how we've defined it. It likes these sort of round gauge-like things. That's the classic way to define what this neuron likes. Here's an epsilon 2.5 perturbation. So that's a very small image perturbations. If you look closely in this dog image, you might be able to see in a strand of hair coming down its nose. There's some subtle changes there. You can barely see it if you squint. You can find those changes. But here's what happens to this individual IT neuron that we think supports the animal's perception when we do this. We drive this neuron up even beyond what it was doing in its clean category preference. So we're able to modulate with that small change to almost flip the apparent category preference of this neuron with a targeted image change. Remember, this is a model-driven image change to produce that effect. And here I'm unpacking that for you as it were different magnitudes of response. The category range is in red. And again, you see with just 2.5 or 5 epsilon units here in the L2 space, we're able to drive neurons beyond the normal category range. So neuroscientists, this is a quite sort of shocking result that these high-level neurons actually have these kinds of effects. That's something we're currently exploring now. The big take home from this that you should take away is that, like, when you compare these things, this adversarial examples, it just gives you sort of one lever into comparing the systems. But if you take these models seriously, they're opening up a whole bunch of scientific questions now. I've only showed you the sensitivity to IT neurons. Perhaps the population as a whole is somehow more robust. Perhaps the brain has other downstream mechanisms that aren't in our current decoders that we need to be thinking about. And perhaps the monkey behavior and the human behavior itself is not as robust as we think it is. We just haven't really done the right experiments to expose its robustness. And so these are ongoing scientific questions. The application question, though, I want to return you to is this one. Regardless of the answers to those scientific questions, this kind of approach has shown that there's an unexpected new way to modulate individual neurons in the brain. And I just showed you, look, a little bit of subtle injection, model-driven, can modulate those neurons quite strongly, many levels deep in the brain. This leads me to my last slide of the talk, really, which is speculate a little bit for all of you. Can we use models to unlock entirely new ways to modulate brain states to maybe improve human well-being? And let me just show you kind of what we dream about on this in this one slide. So idea is, like, now we have these brain visual systems here. We have this whole ventral stream. And what I didn't tell you at the early part of the talk that I'm going to tell you now is beyond the ventral stream shown in gray, the top of the ventral stream area IT connects to all kinds of other brain areas. Some of them are listed here at the right. Imagine, I especially want to kind of focus you on this area shown at the bottom, the amygdala, which is involved in various mood states. It's one of these implicated mood areas. It's probably not the only area that sets mood, but disruptions in amygdala activity are thought to be related to mood disorders. So that's only one synapse away from IT cortex. There's other interesting areas as well. These are memory areas, working memory areas, action areas. These are all sort of other kind of more cognitive function areas here that I'm not going to say about. We've been mostly interested in the amygdala, but the general speculative idea is that imagine that you had precise modulations on your video feed. We're all looking at videos right now that could produce strong modulations deep in your brain, either V4, IT, or possibly your amygdala. Maybe that could lead to gains in health. Maybe they could be better than taking a pharmaceutical to improve your health. Maybe there are ways to do this with digital interventions of this type shown here, driven by modeling. So far, we've shown you that we can do this in a kind of glass half full way, glass half full, half empty in area V4. And I just showed you some new results from IT. So there's definitely some traction here. And as those models get better, again, brain score models, community models will improve. These kinds of, I'll call them superpowers, will get better, and we'll be able to start to unlock these kinds of application goals with better and better models that more and more approximate the biology. So I said that was my last slide. I lied. This is my take home, so summary slide of everything I tried to take you guys through today. So think of system models as scientific hypotheses on systems neuroscience. They make predictions that don't require humans in a loop. The most accurate of those models, the most useful of those are the most accurate. For brain science, accuracy means those that are best aligned with the brain and behavior. I showed you kind of, I gave you a sense of how we do that. The claim is that these are the best scientific hypotheses, the most empirically aligned. I want to remind you, I've said this a few times, there's never one final model. There's always going to be a better and better model. As we get better and better models, we get better and better at our application goals. But I've shown you that even with our current not perfect models, you can start to do things with them, neural control things, for example, that we could not do without those models. So we already have traction, even though those models are not perfect. And a general key bottleneck of our field, this is more of a sociological statement, is that our field has not been very good in systems and cognitive science of shared ways of knowing that we're having better and better models. And there again, I want to re-highlight what we were trying to do in BrainScore as a community effort to help open up this ecosystem and build this ecosystem, at least within the area of visual models of ventral visual stream processing. And we hope that that will expand beyond just visual processing. And we're just one part of that, which really has to be a community effort, not just something that's done in a lab like mine. And we're trying to do our part there. And we hope if you're interested in that, please reach out to me and we can help you be connected to that community. With that, I'm going to end. And if there's any time, and again, I'm happy to stay late and take questions. So thank you for listening.
Thank you so much. We do have a few questions in the chat, so let me go through those. So one was asking, is there a behavioral correlate of that change in IT neuron firing?
That's a great question. So it's sort of when we make these adversarial attacks, does it lead to changes in behavior? So first thing to say is the neural response, when we were, and I glossed over this talk, so much of the neural data we collect is in passively viewing animals that are just fixating. So even though I showed you behaving animals, and it's much easier and more efficient to get data in a passively viewing animal. So we couldn't just say, ask the animal the answer, because the animal we collected that data from wasn't doing a task. In principle, we can do that. And we just haven't done it yet, so I can't give you the answer. I should point out that people have tried to ask whether human behavior is sensitive to these kinds of adversarial attacks, and they've shown some weak effects that there is some sensitivity in those directions. So there's hints that yes, the behavior will be modulated. How much it becomes a quantitative question as to how much it will be true or not. But it is one of the forward-going scientific questions that I outlined in that last slide that I breezed through. So great question. Don't have a quantitative answer for you yet. But thank you.
Great. This is actually a follow-up to the first question. So they said one more follow-up question regarding spiking. Is there a reason for trying to align brain neural models to ANNs, not based on spiking, versus neuromorphic computing engines, which are more based on spiking rates and such?
Yeah, that's a great question too. There's no in principle reason why it wouldn't be better to have models that had spikes in them and align at that level too. There's a couple of sort of in practice reasons, though, I'd say. The first in practice reason is those models, those of you who build those models know they're much more intensive to build. So every one of these models requires a lot of compute to both train it, to learn it, to test it. And so there's as compute gets, and we build, hardware systems that are better able to model that, and many people work on spiking models, maybe even the person asking. As those become more and more available tools for both building and training and testing those models, then that will be a natural path for the field to take. But we really took it more from a practical point of view. The only other, that was one argument as a practical argument. The other one, as I gave you at the beginning, is like the mean rates with decoders has a tight link to behavior. So that allowed us to sort of just say, let's focus on mean rates for the moment, because I didn't need the spike by spike data to make a prediction, a good accurate prediction of the animal's behavior. And if our goal is ultimately to explain the behavior, maybe I don't need a good model of all the spikes. But at some level, the model of spikes will be more valuable. It just wasn't yet for that particular behavioral test. So there's two answers to that same question, which is a good question. Again, I'm not trying to shut down that line of work. I'm just giving you why we started where we did. I hope that makes sense.
Great. Another question. What is your best guess of the source of robustness to adversarial attacks in perception/behavior?
Right. So there's that question presumes, and I'll back up. That question presumes, it's another great question, presumes that we are robust, right? So, I mean, that was sort of the third thing is like, we don't really know how robust we are, which was sort of that's the back to these. What's your best guess? So these are the three possible guesses, right? Well, perhaps monkey behavior isn't robust. This person is presuming that question 3 is, we are more robust than these models. Notice we don't have a full end-to-end engineered model of human behavior. So any estimates we make of humans and neurons, by the way, are lower bounds on our robustness, right? In other words, when people attack these models, they're often making full white box attacks with full knowledge of the model's network. We don't have that for the visual system. So again, I think, well, my intuition is we are more robust. So then it's probably some combination of 1 and 2, right? I'm showing you the sensitivity of individual neurons, but remember, it's more than one neuron driving the behavior. And then somehow they all work together. They don't just go from IT to the animal's choice. There's all kinds of other downstream mechanisms. Even though a linear decoder can approximate the animal's choice, that downstream mechanisms probably also add robustness. So even though I'm telling you individual IT neurons are equally robust to model neurons, the behavior could be more robust in the biology because of these other reasons, which we can't see in the spike rates of IT neurons. So again, good scientific questions that I don't know the answer for. All three of these are basically open questions. Yeah.
All right, another one. In the adversarial example, are the neural spiking increases measured spiking rates for an actual brain? You mentioned this was for the IT region. Does it extend to the other visual regions, which are deeper? Would this imply that even though in machine learning parlance we say imperceptible noise, it is actually perceptible? Sorry, it's a sequence there, but.
Yeah, so again, since this person's asked a question, will allow me to unpack a little bit more about what was actually done. So you're asking about this motivation here of like trying to compare these here. And I skipped through the methods because I knew I wouldn't have time. And I'm sorry if I wasn't clear. We're actually measuring real IT neurons and we're comparing them with model neurons. So how we do that, and if I have time, I'll just show you here in one slide how it's done. So you actually, first you're recording from a neuron in IT cortex. It's chronic recording, so you're holding that neural site over days. You first get some images, responses of that neuron. You can map the neuron into a model in the way I described earlier. Now you can then use the model to design an image under a perturbation budget. And then you have a bunch of clean starting images. So here's an example clean image. Just for those of you who know adversarial attack, these are so-called clean images, which are just the raw images that you start with. Here's an example clean image. Once you have a model mapped to the brain, you can invert through that model, given the perturbation budget on the image space, to find the perturbation that you want to use to best try to modulate this neuron. Here, our target of optimization was to move the neuron's response as much as possible. It was not to make a behavioral flip. It was to move the neural response. So again, that's not what adversarial attack usually does. It usually tries to flip the category output of the system. So here we're just saying, we've got this neuron, please find me an image to move it. So we're using the methods of adversarial attack, which are essentially inverting through a model, optimization with some guidance from a model. We're using that to find this perturbation. Notice the perturbation here. I don't know if you can even see it. You probably can't because it's very small. Here it is amplified, right? So that's the actual perturbation that's used to turn this clean image into this epsilon 2.5 images. And most of the ones I was showing you that have big effects were around 2.5 to 5. So now then we go back and we basically show to this recorded neuron, remember back in a monkey here, both of these images. And we don't show them back to back. We show them interleaved. We show them many times. So we can give you an average spike rate. So you don't have to worry about adaptation effects. We're gonna take care of that. And I don't have that detail here. And then we just measure the delta response, absolute delta response, which is shown here between the response of the neuron to the clean image and the image with the perturbation, which is shown here in the bottom. So I hope this makes sense. So we're then going to get a magnitude effect here. And then we just do this. We don't just do this for this one crab image. We do this for 100 arbitrary images that gives us an average sensitivity for that particular neuron. And then we go back, and in this case, we did it, I say average of the worst case, each case, worst case for each image. Then we go back and do this for 21. We did it in this case for 21 recorded IT sites that we happen to be on recording at the moment. But again, then we can average over many neurons, and that was sort of the result I was showing. So we're giving you kind of an average of averages sensitivity here, if that makes sense. Hope I answered the question, and Mark, please tell me if I wandered off of it. But basically, there's one more bit you have to do, which is the units of this are spikes per second. The units of the model, we mentioned this earlier, are activations. So we have to normalize when we compare one to the other. We normalize by the natural response range that gives us a dimensionless quantity to allow us to make that head-to-head comparisons that I showed you on this slide here, which is what's on that XY axis here, is a normalized sensitivity comparing biological and model neurons once you make all those normalizations. So anyway, thank you for giving me a chance to show the slides I cut out. Yeah.
You're good. I'll get back to the other questions. I'll just follow up with this one since it's, I think, related. So this person says, a tiny perturbation of stimulus strongly modulates neural firing rate. What's the underlying neural mechanism? Feed forward, feedback, local disinhibition, ... Any guesses?
Wow, everybody asks these great questions. They should come to the lab and do this. We don't know, right? So this is I guess it's sort of like the model says this is what should move it. Now, that maybe raises a question of like the model also predicts when we make these movements, what's happening in the model's V4, what's happening in the model's V2. Like we weren't testing those predictions, but the model, by choosing this image, is choosing other things to tweak in the system. So those are the model-based hypotheses. These models here, these are feed-forward only models. So to the extent this is lining up with the biology, it would suggest that we're just putting, the way I think about this is we're putting energy on the retina in the right way that unlocks nonlinearities up the visual system and the feed-forward path to produce these responses, because that's all the model knows about. The model's a feed-forward only model. Now, but maybe it's capturing what are essentially recurrent biological effects inside the feed-forward processing of a feed-forward architecture. We can't say that that's not true. Remember, a recurrent model can always be unrolled into a feed-forward model. We're just comparing a feed-forward model against the biology right now. So the answer again is we don't know, but I'm guessing most of these are effects that are feed-forward effects in the visual system. But that's a hard question to actually answer, because you'd have to do silencing of the recurrence in the biology and things like that to sort of start to tease that question apart. But maybe the models will offer predictions along those lines that could be more directly tested, that intuition.
All right, this question from Cynthia. Which tests are the hardest to pass, as in approaching neurological responses? Do you have guesses based on the biology why they may be difficult and require adding in other information like spiking information or oscillations? Kind of continuing, have you compared the prediction of the mean rate model to the predictions of the spiking models for what stimuli would lead to certain neural responses? Do they align? There's a lot there.
Oh wait, so there's,
and you can see the text in the chat if you want to look at the
oh
question.
yeah, let me sort of go. I can't see my chat, but there were sort of two questions in there. So let me see if I can, they were sort of different questions. So the second, let me see if I can do the first one was essentially where are the models least aligned with the biology, I think, or something to that effect. And again, I mentioned this BrainScore platform. I'll zoom back to it. I'll put that back up. So if you guys can see that, I'm going to shut this format window down, or here, let's get rid of that. Anyway, try to make this bigger. Well, anyway, I hope you guys can see that. So here, this is a collection of benchmarks. So they're organized by brain areas, like V1 has a whole bunch of them, and V2 and V4 and IT have less of them. We're still building up this platform, so it's hard to know when you say which are the least aligned. We could look at these numbers and say, right now, if you just went BrainScore site and downloaded them, you'd find that the V4 alignment, I think, is the weakest alignment. But that might not answer the question of which neurological thing, right? This is just which parts of the brain are least aligning with which neural data are least aligning with the models. The question might have been a little more at the behavioral level, which kinds of behaviors are least predicted. But maybe the question wants to be asked again, so I can answer it because I'm reading it. Which tests are hardest to pass? Yeah, I see. So again, these are all the tests here, and I wish I should have this is a sort of, I don't know. I actually don't remember the answer to that question. But if you went to the BrainScore website, so you could try to answer that even looking at the BrainScore. One thing I want to say here is that question itself requires a good estimate of the ceiling of what past even means. So we can score models on how well they're predicting these things, but to answer this question requires a fair comparison across the benchmarks. That's actually a very challenging thing to do because it requires a commitment to what a ceiling means. And there's different versions of a ceiling. So it's something we're working on with this platform right now. So that's partly why I don't want to, even if I went to the BrainScore website, I wouldn't want to give you an answer for sure and say it's definitely this is the weakest prediction. And keep in mind, the weakest prediction will always be with respect to the quality of data that's obtained experimentally, which is itself a subjective call of the field, right? So it's sort of that question is it sounds straightforward, but it's actually a very difficult one to answer in a quantitatively accurate way. I guess I'd say they all look half full, half empty as a sort of a short form answer. So there's not one that jumps out at you of like, this is obviously wrong. That's the short form answer, which is I wish there was something that popped off the screen, but there's nothing like that, or I would be talking about that. And
Cynthia, do you want to jump in?
Yeah, something about spiking networks again that I didn't answer yet. So I'm sorry, Evan. Maybe you could answer or say again.
Yeah. Let me see. Cynthia, do you want to jump in, or should I just go ahead? Okay.
Oh,
go
ahead.
Yeah, go ahead, Cynthia.
Oh, you meant to follow up on the last question, or?
Whatever you want to talk about.
Okay, thank you. So yeah, I think you're hitting on it. I guess I was curious about those tests because I know there's a lot of like working memory and attentional visual tests that are really popular for studying things like oscillations and synchrony is thought to be important. So I think that's where that question was coming from. But I also know that there's a lot of spiking models ongoing. So the other question was just, have you kind of compared predictions of spiking models for what might be causing strong perturbations to your models and seeing if they actually agree essentially on what we should be doing to interact with these regions.
Okay, wait. You said three things, all of which are interesting. The first thing I'd say,
Sorry.
like, when you say your models, these are the fields models. So
Sure.
we contributed some of them, and you can see, you know, the people building them, the names are on the left. But this is really, I think, a community effort. So the fields models, like, where are they weakest, I think is your question. So
Absolutely, yeah.
and then you also said the other thing you're interesting and important, you said, is like, you know, attentional effects, oscillations. This platform doesn't most of that's not yet up on this. This is really kind of, think of this as just feed forward approximation models is really where this started. So there's an effort to try to expand this in various ways and to add more behavioral tests. And, you know, Gabriel Kreiman's lab is one that's done some work. He hasn't yet got that on BrainScore, but if you're interested in those kinds of attentional search effects, that's a lab I would point you to some of their work most immediately. It's just not in a community-oriented format to sort of compare that, and with many models, at least not that I know of yet. And we're working with Gabrielle to try to do that going forward. And then the third thing you said was the spiking, like, how would you compare spiking networks with these networks? So that actually is a question that kind of comes down to, like, none of these benchmarks require spiking. They're all like, these are all mean rate prediction benchmarks. So we'd have to start to add spiking level benchmarks onto the platform. And then many of the mean rate models would, of course, fail miserably because they don't make individual spike time measures. So it requires some effort to someone to commit to starting to put up those kinds of benchmarks on these kinds of things. And so that hasn't been done yet. I might turn it around and ask, why do we think the spike rate models are the most important to do, right? So there's that, we sort of avoided it for the reasons that came up in the earlier question. But it was really a subjective call, right? And a practical call, as was mentioned earlier. Not fully subjective, but again, because there's always a next question. Well, what about the synaptic prediction strength metrics, or there's always a more detailed model, so to speak. So what the right way to stop is an unclear question as well. And these platforms, when you build it, exposes that, hey, there's lots of ways of biology we could make to be successful, and nobody really has a corner on that market of the right answer. I certainly don't, but I just kind of, you know, say that those are choices that have to be made in practice. They're made by whoever wants to put in time to put up a benchmark on a platform. Is what I'm hoping will be the way it's going to actually unfold in the field. Does that make sense? Or at least if you have thoughts on that too, I'd love to hear them.
Yeah, no, that was all really interesting. Thank you.
Yeah. Mark, you're muted if you're.
Oh, yeah, thanks. Yeah, so this question: for a model specified to align IT neurons, how good are the layers prior to that V1, V2, V4 aligned to their biological counterparts? Is there any data for that?
I think, yeah, I glossed over this in the talk. So what I tried to say is these models align multiple levels. So it's not like one level of the model aligns with all levels of the brain, right? So one of the first results out of the models was that 2014 result was that the high level of the model aligns, that the penultimate layer of the model aligns best with IT. One of the intermediate levels aligned best with V4, which is the input to IT. So that kind of fell out of optimizing deep networks to doing visual tasks. Others later showed that the early levels of the model, so-called V1 of the model, aligns best with V1. But there is still this art of like mapping areas of models onto brains. But if you take any model and scan it, let's say you scan it, each of its layers of a deep network aligning with, say, V1, you'll find that it's best in the earlier part, but these, and worst in the later part. And the same for you do the opposite for IT. It's best in the later part of the model versus the earlier part. So you can generate curves from that. But these curves are not delta spike functions. They don't like, oh, this layer is actually IT, right? It's like the V4 can kind of look like IT, and the IT can kind of look, the areas that are close together are hard to separate and say that definitely should be model area IT, and that definitely should be model V4. So to really score these models requires the modeler to even make a commitment of what is it going to say as its IT. So I'm just pointing that out that yes, I think the answer to your question is, do the early levels align? The answer to that is yes, they align pretty well, but not perfectly. The same half full, half empty answer I've been giving you for almost every one of these questions. But also to point out that aligning models with all levels of the brain is itself one of the challenges that is sort of skipped over and how that's usually managed. But just to say, it's not, you should keep in mind, it's not like one area of a model fits everything. That's definitely not true. I hope that makes sense. And please ask follow-ups if it's still not clear, because I love the discussion. I'm happy to stay.
I have a question if no one has anything right now. The terminology may not be exact, but you referred to basically the modulation of like the preferred neurons and the non-preferred. What was the terminology used? Basically, the non-preferred kind of was modulated more by the non-preferred. Yeah.
So and again, I'm sorry I was going faster because I knew I was out of time. So remember, this is one neuron, and the way if you record from an IT neuron, people usually say, "What's its preferred category?" You know, oh, it likes faces. Oh, it likes cars. What, you know, and the way they assess that is by scanning a bunch of images from categories that have been predefined. Like these are like ImageNet categories that we used here. I forget what this is called, valves or something. And then this is some class family of dogs. So then you just go ahead and measure the mean rate, and whatever its highest category averaged over images, you call that the preferred category, and the lowest you call that the least preferred category. And that's essentially what we did here. Yeah. So then when I said what we did was we can do the opposite trick. In fact, here we did the trick of taking the low response and tweaking the neuron to make it activate. So we started, you think of this starting from clean images, which for this neuron happened to be less preferred category, updating them individually to be more activate the neuron. And so I'm just doing this to contextualize the magnitude of the effects that the perturbation caused. We actually did the opposite. You can take the preferred ones, I just don't have it here, and make them try to modulate the response downward, and you can do the same thing. So you can take the red things and make it go low by modulating the response. So the key point is that you can move the neuron's response quite far with a subtle modulation of the image that the model's directing you towards. I think the upward effects were slightly stronger than the downward effects, or at least our ability of the model to find them so far. And so there may be something interesting in there. This paper, by the way, isn't even published yet. It's under review and so forth. So this is all still early. I'm showing you like preliminary results. So when that paper comes out, then, you know, there's 12, you guys are all asking great questions about what this means and follow up. I thought I'd show you this because I thought it was quite interesting, but I'm sorry I had more questions than answers on all of this. But Mark, did I at least answer did I clarify what your question was here, or did I not?
Well, I guess that was the foundation. So I guess what my question was related, and I know I'm probably using maybe a loaded term in terms of like salience, but is there any idea about, like, you have these big perturbations or changes in the spiking rate, and we're presuming that we are still biologically able to perceive correctly between the categories and overcome, even at the low level there's a change, but at the high level we're still able to understand the image. So the question kind of is there any kind of idea about how this large change in spiking rate is able to be neglected, where the robustness comes from? Is it some sort of like salience based kind of like, oh, we can ignore this big change in the spiking rate because it's not actually, doesn't matter to the overall image understanding that we want to achieve? Or I don't know, what's kind of a hypothesis there?
Well, okay, so here's a thought experiment to help keep in mind. Suppose I was doing this on your retina. So I'm recording your photoreceptor. I'm recording a photoreceptor, and I'm showing these images, and I figure out I'm going to tweak this one pixel to make it a little bit brighter or darker. That's going to have a big effect on your response of that let's say retinal ganglion cell, to make it spiking, because the other ones all spike. And that, like, okay, and now you're like, oh yeah, well that's obvious. It's like one pixel, like, one pixel still looks like a dog to me, right? So you're like, okay, then this result wouldn't be surprising. The only reason it's surprising is like these are high level in the visual system. So like people often think is that they're much more tied. There's evidence to show they're more tied to actual perceptual report, like things like binocular rivalry. When you're perceiving, these neurons modulate up and down, whereas your retina does not. So these are more associated with your reported percept. So that begs, so again, I'm just saying, if you just think of it like the retina, you're like, well, this is like, you got neurons, you got an end, you got a model, you modulate. It's like but you're raising the kind of the linkage to perception is the question you're raising. So how and really it's like the way we used to do linkage of perception was you have a linear decoder linking these neurons to the report. That's the thing I introduced at the beginning. notice this is still not inconsistent with that idea because I'm only modulating one neuron about, we estimate it's around 50,000 neurons would be the right number of neurons to estimate, kind of get the actual trial by trial performance accuracy with a linear decoder. So it's like 1 out of 50,000. It might be like, a drop in the bucket here, but that presumes that when we modulate this neuron, all the other neurons are not modulated at all. And they probably are modulated because we don't have control of all of them. That's back to that modulate one, 4999990. We didn't do that experiment. It's not really doable today. But again, these modulations might be along axes which are orthogonal to the category boundaries. So you still perceive it as a dog, but your visual system is being driven. That's the one I prefer. It's just because I can modulate neurons. There's lots of directions in image space, which we just normally don't encounter, right? So it's like, this is like the model. This is a worst case scenario of the models found this thing, right? So, but remember, I should make a plot of like, if you just do random search, it's like unlikely to have these kinds of effects. So your visual system may only be robust to the level at which it needs to be. And that's kind of back to the speculation I made at the end. It's like, there may be, just like pharmaceuticals were designed to take advantage of things that biology left, like opioid receptors, right? Like those were left over from biology. There may be holes in the visual system biology that didn't need to be plugged, but are available for modulation. So you could have orthogonal modulation of your visual system that finds its way into your amygdala, has strong effects, but doesn't affect your day-to-day life. Like that's the thing I'm dreaming about right now, right? So because you're basically assuming that this neuron has got to drive the perception. I'm like, well, yes, but it's not clear that those are science questions still unanswered. I'm telling you, even with our current models, it might still all hold up that this is not enough. I don't know the answer to that yet. But those were those 3 things at the end that, you're asking, how does the system become robust? I'm like, I'm not sure it needs to, I guess, is a form of the answer. Again, given the decoder models, this is not out of line with that idea because I'm just showing you one neuron. So that's, and we have to think that through, work that through, try that out. I think what's going to happen is going to be some combination of both, all of them. First of all, it's actually, you didn't need to model the model. It's all 3. It's like, it's a single neuron. It's the linear decoder is actually not the right decoder. And you got to think of the whole population and the behavior is not as robust as we think it is, right? So it's like all 3 of those is going to triangulate to the truth. It's not like one of those is the answer, right? So if you follow what I'm saying, it's like these 3 questions all answer yes. So if I go one more, let's see here. Okay, this is one neuron. You think of the population, it's more robust. Linear decoders are probably not the right decoder mechanism, which relates to this attentional things and things you were using, this like more advanced decoders of how the neurons give rise to the percept. And then the third thing is like, well, okay, we're not actually quite as robust as we think we are, right? So I think some combination of all of these 3 is going to be the truth, not one of these 3. So your question was phrased like, let's assume 3 is true, and then the problem's got to be in number 2 was sort of my phrasing of your question, right? So, but I'm also channeling 1 and 3 at the same time. Does that? I mean, sometimes these are logical space of possibilities, but I'm saying all of them are probably partly true.
Yeah, I think that's fair. Great. Okay, I think we have one more question. If you made an adversarial attack with respect to the output of a linear decoder, you might expect more of a perceptual/behavioral change, I guess.
Yeah, and that was the first experiment we were actually thinking about, but we decided to go with this one because it was sort of easier to do in some ways. But yes, we could try to make more direct adversarial attacks, but yes, we just haven't done that yet at the behavioral level. This is back to the earlier question. I mean, we wanted to just we thought this was a thing that we had more leverage on as experimentalists in IT. It was where we're sort of trying to make claims about whether IT neurons are as robust. So we started with this one. But again, I'll emphasize, this is not a standard adversarial behavioral attack. This is just a neural directed attack, which I like it because it has these neural control kind of payoffs in it, but it's not. The other proposal was on our table. We just didn't do it first. So I think we will get to that. I hope at some point, or someone will get to that, maybe motivated by these results. But yes, you guys are asking the great follow-ups again, and you got to get me back in a year or 2, and I'll hopefully give you some more answers on that.
Great.
Or if anybody on this call wants to come work in the lab and help do this, like, please just ping me afterwards. I put my email up at the end. Always looking for good students and postdocs. So I'll do some. I'll put my email up there again.
Great.
Do some shameless advertising. So
no worries. Well, I don't think there's any more questions. Is there anyone else in the audience who wants to ask questions? Any questions? Okay, great. Well, thank you so much for joining us. I think we all learned a lot. So thanks for your time.
Yeah, thank you for having me. Great to be back virtually for some time, guys.
All right.
Talk to you soon. Thank you, Mark. Take care.
Yeah, no problem.